Good afternoon. Welcome to the Department of Medicine Research and Innovation Conference. Today we have two speakers. The first is Dr. Joshua Barocas. Joshua did his undergrad at the University of Colorado and finished at WashU in St. Louis, graduating cum laude. He did his MD degree at George Washington and was AOA. Then he did his residency and chief residency at University of Wisconsin, and an ID fellowship in Boston at the MGH in Brigham. Joshua joined the faculty after his fellowship, joined the faculty at Harvard as an instructor, and then moved to Boston University as an assistant professor and director of infection control and research. In 2021, he moved to the University of Colorado, where he's currently an associate professor of medicine. Joshua is the PI of two NIH grants, and his area of research is using clinical epidemiology and mathematical modeling to inform decision-making and policy related to improving health outcomes of people with substance abuse or substance use disorders who are at risk for infectious diseases. So Joshua, why don't you go ahead?
Absolutely. Thanks for having me. I will go ahead and share my screen. One moment. So briefly today, I'm going to just be talking about some of the work that I've done with novel approaches to estimating the scope of the overdose epidemic here in the United States. I purposely call it the overdose epidemic because at this point, we all know it's beyond just opioids. As was mentioned, I'm supported by the NIH, specifically NIDA on two awards, a DP2, as well as a KO1, and I'm a co-investigator on a U grant. I usually start out my talks with a quick land acknowledgement, acknowledging that I'm giving my talk on the ancestral lands of the Arapaho Nation, and I'd like to acknowledge that. All right, so we know that overdose is a big problem in the United States. Current numbers show that in 2020, approximately 100,000 people in the US died of a drug overdose. The majority of those deaths, though not all, but the majority of those deaths are opioid related and occurred in people with opioid use disorder. Here on the right, you can see the provisional number of drug overdose deaths over 12-month periods that just keeps on going up. This was supplied by the CDC. Now, as I said, most of the overdose deaths are among people that have either opioid misuse or opioid use disorder. We need to know if the overdoses are just going up or if the size of the pool of people at risk is also going up. So we need accurate estimates of opioid use disorder at both the state and national level. We need this not just for our own understanding, but specifically to inform resource allocation, service planning, targeted interventions. We need to do this to decrease health disparities. We're only recently sort of skimming the top when it comes to health disparities when in the overdose epidemic. And just fundamentally, we can't reach people who we don't know exist and are at risk. And I say all of this with sort of the backdrop that we have a gigantic failure of what we call the gold standard approaches to estimating both opioid use disorder in general, but also drug use. So we use direct estimation. That's sort of the what we would consider the gold standard. And this comes in two forms. One is through survey data, and two is through administrative claims data. Survey data, like the National Survey on Drug Use and Health, is problematic for a few reasons. One, it excludes large swaths of the population, specifically large swaths of the population that we might be interested in helping find people who are experiencing homelessness, people who are incarcerated, and people who are just hospitalized. Also, because substance use is so stigmatized. There's these surveys, which are household surveys where you answer the question in front of somebody else, are subject to social desirability bias. So people, you can imagine, may not want to share their substance use with a stranger. Second, as I mentioned in this, in how we do sort of direct counting, is with administrative claims data, EHR, Medicaid claims, etc. Now, of course, this requires somebody to have touched the healthcare system, and for someone like me, a physician, to have both appropriately and accurately diagnosed me with opioid disorder. So you can imagine there's a lot of ways that this fails, that this approach fails us and undercounts our prevalence. So for the past couple of years, I've gotten really intrigued by using indirect estimation for opioid use disorder prevalence, and hopefully we'll be able to move this along beyond opioids. There are two basic ways that we've been approaching this. One is through a capture recapture method, which I'll talk about a little bit more in depth than the other. The other is through what's called a Bayesian hierarchical multiplier method, which is a lot of words, but essentially I'll do the easy version of it today. And we've been doing this both in Massachusetts, Kentucky, and Ohio. Now, this is, some of you may have seen this slide before, but I love going through this because capture recapture is an epidemiologic method that is actually was started through Fish and Wildlife. So we're here in Colorado. I should probably make this how many elk, but essentially how this works is it's predicated on basic rules of probability. At time one, you're out in the woods and you capture 60 deer and you tag them on the deer. You let them go. And then at time two, you capture 30 deer. You see how many of those have actually already been tagged. And based on just sort of basic laws of probability, we can figure out the total population of interest in a given area is approximately 120 deer. Now, of course, humans are very different than deer. And so we have to come up with different ways of using capture recapture approaches and put a lot of restrictions on it. I could talk about this for the next 3 hours. What I'm going to do is sort of the brief overview of the analytic steps of what we do in capture recapture to understand prevalence of opioid use disorder. We take a number of different databases that have individual level data, not aggregate level data. We identify variables within those data sets that are indicative of opioid use disorder or whatever disease or disorder or condition we're trying to get at. We construct these gigantic contingency tables, oftentimes stratified by things like age, sex, and county. And we construct multivariable statistical models to estimate the unknown population.
Joshua, well, let me interrupt for a second. We've had a few people in the chat box say that the sound keeps coming in and out for them as well, not just for me. And I wonder if it has something to do with your microphone that you might be able to fix. I don't know.
Let's see. Is this better?
Yes, it sounds better.
Well, we'll do this then. That's
okay. Thank you.
Apple has failed me in my use of their Apple Pods or whatever these things are called.
All right, that is much better. And
all right, great.
Also, let me also say that if people have questions during the talk, put them in the chat box or in the Q&A, and we'll get to them afterwards. All right, sorry for the interruption.
No, I apologize. Sorry about that. Thanks for interrupting. All right, so in brief, we construct multivariable statistical models, estimate the unknown population, and we use bootstrapping methods to estimate confidence intervals. And of course, we end up conducting a ton of sensitivity analysis because, as you can imagine, there's still quite a bit of misclassification. There's quite a bit of a discrepancy in how we want to define variables for opioid use disorder. So as I said, I can go through these steps with anyone that's interested in just painful detail. But for right now everyone is at least a semi-expert in capture recapture methodology. So the first step is to have linked data sets. I've been very lucky to work with both the Massachusetts Department of Public Health and the state of Kentucky, where there are two large data warehouses that are linked at the individual level. These, in Massachusetts, it links data at the individual level with 25 state agencies, and we identified 7 of those databases where we could accurately, or what we think is accurately, define a variable for opioid use disorder. This, of course, is claims data, hospitalizations, substance and addiction services, death, birth, where, and by birth, we can find infants who have neonatal abstinence syndrome and link them to their mothers, EMS records, and the prescription monitoring program. The same sort of magnificent warehouse exists in Kentucky, where we've used 4 data sets, Medicaid, death, EMS, and the prescription drug monitoring. So you can see there's what ends up happening is where, for instance, Medicaid may undercount in and of itself because we can find somebody in an EMS record or in a prescription monitoring record or in a death record. We can figure out how many people or where that person might have been quote unquote caught in a given year or in a given time period. And then based on interactions, we can estimate the unknown population. So what have been our major findings? Well, back the first iteration of the Massachusetts data set included data from 2011 to 2015. It's been since expanded and we're working on newer updates. But what we found and what led us down this very long trail is that as of 2015, what you can see is in this bottom line here, is that we could count 119,690 known individuals. That means that someone showed up in one of those 7 data sets with opioid use disorder, or that nearly 120,000 people showed up. How they showed up and in what combination of data sets helps us get to this unknown number, this 155,000. Giving us a total of 275,000 people with presumed opioid use disorder in Massachusetts in 2015, which is a prevalence in the overall population over the age of 11 of 4.6%. Comparing that to what the National Survey on Drug Use and Health estimated of 0.94%, we're calculating that NSDA, which the shorthand, is likely underestimating Massachusetts prevalence by 5-fold. The same sort of thing we found in Kentucky. So this is 2018 and 2019 data. 2018 here in the gray, 19 in the white, second row. What you see is that our known population, people that showed up in one of those 4 data sets, range from about 92,000 and 98,000. We were able to estimate somewhere between 56 and 61,000 people, and getting us a prevalence of between 5.45% and 5.9%. Again, comparing to the National Survey on Drug Use and Health in 2019, they said 1.3%. We said 5.87%. Again, a 5-fold underestimation from NSDA. Just striking how many people we're probably missing that we just don't allocate resources for. So I'm going to move very quickly through what's called a Bayesian hierarchical multiplier method. And this will be the only equation that I show today. But essentially what we've done is we've utilized just 2 data sets, the all payer claims data and the vital statistics records for overdose death. And essentially what we do is it's a fancy multiplier method. So all you do is you say, what were the number of opioid related deaths during a year? And that's the product of the rate of opioid related death for people that we can see in the all payer claims that have a diagnosis of OUD, and the total number of person years at risk of opioid related death. So you multiply those together. So essentially we're using known data to estimate the unknown OUD prevalence. We've done this in both Massachusetts and Ohio, and I think that we can do this here in Colorado. I almost said Massachusetts, didn't I? I think we can do this here in Colorado. It's a matter of linking our Medicaid data to our death data. What I want to show is when we did this in Ohio, we did it in a Medicaid population in the 19 counties that have been most impacted by the overdose epidemic. For instance, Scioto County, you may have heard of because it was referenced in the book Dreamland. The bottom line here, the bottom line finding is that our naive estimate, so when we just look at the direct observation of OUD diagnoses among people 18 to 64, we see a total population with OUD of 53,000. By linking this to the death data and doing some fancy math that I did not go into, we actually found that the estimate was around 13% for all of these counties. Now, you might be sitting there saying, geez, how do you know that either one of these is true? These seem high. I'm not sure that I believe you, Josh. So what we did is we actually took our capture recapture method, where I said it was 4.6%, and we did the same thing in Massachusetts on the same for the same year. And we took that Bayesian hierarchical method. What we found was an overall prevalence of 4.29%. So we're within spitting distance, and it provides us with a little bit of validity that our estimates, though they may be high, we're certainly way off when it comes to the National Survey on Drug Use and Health. Now, oops, sorry about that. Now, it's clear, as I just said, that relying on these single data sets or surveys is just insufficient. What we need is linked data at the individual level. We've been doing this, as I said, in Kentucky, Ohio, New York. We've started some of this work, and in Massachusetts. And soon I hope to be able to take some linked data in Colorado now that I'm here. And just to give a sneak peek, because I know that one of the big things here in Colorado is stimulants. So we actually started doing some work within the stimulant realm, and using first and foremost this capture recapture approach. We have 2015, 2016, 17, and 18. And what you actually see, like I said, these are very preliminary results. But as of 2018, we found approximately 45,000 people, just without even any estimation, just by looking for people in multiple data sets across Massachusetts. We found 45,000 people with what we're calling problematic stimulant use, so misuse or use disorder, which is roughly about percent of the population. Now, just for comparison, the National Survey on Drug Use and Health says that the prevalence of stimulants in the U.S. is about 4% or 0.4%. So even without doing any fancy math, we're finding that we're undercounting. So here are my takeaways. One, thank you for your attention. I love talking about, and I think it's crucial that we talk about how we're missing populations. And for anybody that was wondering, my daughter wanted to paint my fingernails before my talk. So I have I have watermelons on my fingernails, and if you were curious. I believe that this has to be done with active engagement with state government officials. This couldn't have been done without that in Massachusetts and Ohio and Kentucky. I believe very strongly that this happens when we share code, when we have linked data, and we have involvement of people at the National Institutes on Drug Abuse. And finally, I'm not sure where I am on time, so how much time we have for questions. But I just want to say that every overdose death is a policy failure, and we can certainly do better. I'll stop sharing at this point.
Thank you very much, Josh. We have a question in the chat box, if I can get to it here. Yeah. From Susan Calcaterra. She wants to know if the national survey does a better job of detecting problem use compared to measuring disorder. What do you think about that?
So I think that when we look at it's a great question, Susan. When we look at data on how people actually share their substance use disorders, whether it's alcohol, marijuana, or opioids, people are largely not comfortable or underrepresent their substance use. I think that where the National Survey on Drug Use and Health specifically fails is not necessarily in respondent and in that sort of social desirability bias, but specifically, I think that it fails because it does not account for people who don't have a household. So where we are, especially when we talk about healthcare disparities and marginalized and vulnerable populations, the household survey for NISDA does not account for doesn't look for people in jail, prison, hospitals. So even if somebody knocks on the door and you happen to be hospitalized or in the emergency department, they miss you. And so I think that they continue to undercount even with problem drug use.
Another question from Ed Janoff. What proportion of overdoses occurred in the ascertained drug users?
Ed, this is literally the $1 million question. So the way that our methods are set up, specifically for the Bayesian method or the Bayesian model, we define the population and make an assumption that all overdose deaths, all opioid overdose deaths occur within people with documented opioid use disorder. Now, we've done some sensitivity analyses that'll be forthcoming in the Ohio work and hopefully in the Massachusetts work as well. And what it shows is that no matter how you define OUD in that first group, in the known group, we end up still getting a very, very high prevalence. So I can't tell you exactly what proportion of overdoses occurred in people who use sort of regular drugs or have use disorder because of how we defined it. And we're figuring out some methods to sort of get around that.
Mark Ernest wants to know what the limitations are to replicating this in Colorado.
So the only limitation at this point is data. So we have the methods, we have a very good algorithm that is replicable in all of these various states. At this point, we need partners who are able to link data for us where we can go in with our algorithm and estimate. And I know that some exist. It's been a matter of making sure that the algorithms are correct and that we believe them. And it's a matter of working with some of those data partners.
Eric Campbell suggests that you can use your methods to estimate frequency and then reanalyze a random sample of the respondents.
Eric, you're right. We actually we could, and for some of the modeling, some of the agent-based modeling work that's coming out of New York, that's exactly what we did. We used a very crude approach to that for that. That manuscript is under review as well. We did it sort of back of the envelope, but you're absolutely right. And I think that that's where people are hoping some of these methods could go. Alternatively, Eric, I think that even SAMHSA, who administers the National Survey on Drug Use and Health, admits that that a new approach is needed. And this provides us with having all of these various approaches. A toolkit provides different states with different data structures, different data use agreements, and different investment and resources to get some of these estimates done. It provides them with the ability to do it without having to have this massive linked data system like we have in Massachusetts. And I'll answer. I love. I just saw Matt Taylor put in. I'll come back to Ed's in a moment, but I just saw Matt Taylor's question, which I think is a really, really important one. Real-time estimates are really important when it comes to overdose and opioid use disorder and any drug use disorder. How we define real-time has to do with who the end user is and who the stakeholder is. So for some of our work, budgets are made on a yearly basis. So we're able to provide the stakeholders in Massachusetts and in Kentucky and Ohio yearly updates for their estimates, which then inform budget allocation. I'll say when we did this in Massachusetts, we published our 2015 data. It was obviously a little bit delayed. It was the first time that anybody had done this, but it went directly. It was one of the most nerve-wracking things that's ever happened to me, aside from, my wedding and birth of child. But the report that we put together literally went from my hand to the governor's desk, and within 3 weeks, the governor had allocated 4 times the amount of funding for the opioid epidemic in Massachusetts based on the estimation. So real time has to do with who the end user is.
And Ed Janoff had a question before that. Once the population at risk is identified, what do you do to then limit the overdoses? Now that turns you over into treatment, and that's a whole different kettle of fish. Absolutely,
and I think that Ed, you're speaking my language, because what we're getting at is we have to figure out not just what the size of the population is, but exactly who we are probably missing. One of the problems with using sort of direct estimation is it's the lowest hanging fruit. You're going to find the people that you're going to find. And in this case, what we actually can do, and we're doing this work with stimulants, is we're trying to estimate the unknown population in what we'll call vulnerable or marginalized populations, communities of color, people who are experiencing homelessness, incarceration, etc., where we wouldn't have otherwise counted them. So if we know the size, and we know the size in marginalized communities that we've probably been undercounting, then the next step is getting the evidence-based interventions to the community.
Okay, Joshua, thanks very much for a stimulating presentation. Enjoyed hearing what you're working on. Thank you very much.
Thanks for having me.
Our next speaker this afternoon is Makoto Miyazaki. He did his undergrad and master's degree at Showa Pharmaceutical University in Japan, and did a PhD at Nagoya City University, all in pharmaceutical sciences. He did his postdoc at the University of Wisconsin in biochemistry. Makoto joined the University of Colorado. Following that, he's currently a professor of medicine and he has appointments in the divisions of nephrology and endocrinology. He's the PI or co-PI on 3R01, and his area of research is lipid and mineral metabolism in the regulation of obesity, diabetes, atherosclerosis, and vascular calcification, and also identifying novel factors that contribute to lipotoxicity, phosphotoxicity, vascular calcification, atherosclerosis, obesity, and diabetes. He's got publications in a number of high impact journals, including Science, JCI, PNAS, and the Journal of Biological Chemistry. So Makoto, take it away, please.
See, let me share my slide.
Yeah. There you go. Okay.
Okay. Yep. Thank you for generous introduction. Today I'm going to talk about a project that we started a couple of years ago in collaboration with the Dr. Judy Blaine in our divisions. Basically, this project related with cardiovascular complication and dysregulation of phosphate in CKD, and as well as the aging. And this project has been funded by NIDDK. So past 15 years since I started my lab, we really interested how the CKD induced CVD cardiovascular disease, because CVD is the #1 killer for the CKD patients. As you can see that kidney decline of the kidney function is highly associated with cardiovascular events, and especially the patient with less than 15 GFR, like ESRD patient, about 40 times higher cardiovascular risk than a patient with normal kidney functions. Among several cardiovascular complications in CKD, we really interested in one of the cardiovascular disease called vascular calcification. And the vascular calcification is a disease that depositing a lot of minerals in the blood vessels. So this is a micro CT analysis of CKD patients, and this deposition of the calcium in the blood vessel basically losing the flexibilities. So vascular calcification is highly associated with the mortality of the patient with CKD, so it's really deleterious for the CKD patients. So the patient with more severe calcification died earlier than the patient with the less severe calcifications. So our group thinking that two major toxic pathways contributing to vascular calcification. So first one, lipotoxicity, toxicity induced by accumulation of the toxic lipids like cholesterol and fatty acid metabolites. That's one of the another projects that we are working on. But today I'm going to talk about phosphotoxicity, toxicity induced by high phosphate, because that hyperphosphatemia, like increased serum phosphorus, is another major complication in CKD and really associated with the prevalence of the vascular calcification and other cardiovascular complication in the CKD. So I'll talk about phosphate today. So phosphate is the essential element in any cells, because the phosphate is a major source of the ATP synthesis and the intracellular signaling. But when that really high level, really toxic, and phosphate is also the major determinant of lifespan of any mammalian species. This is a slide shows the relationship between the serum phosphorus level and the lifespan of the 18 mammalian species, and number 17 indicating humans. Number 2 is a mouse that I'm working on. So human, we have around 3 to 4 milligrams per deciliter of the phosphate, and our lifespan is around 70 to 80 years. So mouse is a shorter lifespan, about 3 to 4 years, but they have more higher serum phosphorus, 9 to 10 milligrams per deciliter. So as you can see, the serum phosphorus and the lifespan have a very tight correlation and negatively correlated with each other. So phosphate, high level of phosphate is really toxic. Phosphate level is regulated by two major hormone, PTH and FGF23, and kind of pretty simple when in terms of phosphate homeostasis. So dietary phosphorus, like soft drink, a lot of phosphate, and absorbed through the intestine, through the intestinal phosphate transport, NAPI2B, and absorbed phosphate distributed to all organs for the synthesis of the ATP or skeletal genesis and intracellular signaling. But when the two major tissue, parasite glands and the bone, sense the high level of phosphate in circulation, these glands secrete the hormones. So parasite glands are secreting PTH, bone secreting the endocrine hormone called FGF23. So these endocrine hormone, also related hormone, PTH and FGF23, selectively act at the kidney, especially proximal tubular cell, to regulate urinary excretion of the phosphate. So kidney is a central tissue regulating serum phosphorus. So PTH and FGF23 downregulate NAPI2A and NAPI2C, which is kidney-specific phosphate transporter. This protein is reabsorbed phosphate from urine to increase serum phosphorus. So these are downregulate this NAPI2, endocrine hormone downregulate NAPI2A and increase urinary excretion of the phosphate. So this is a PTH signaling. PTH signal is relatively specific for proximal tubular cell of the kidney, because the PTH type 1 receptor is very abundantly expressed in the proximal tubular cells. So PTH bind to the PTH type 1 receptor and activate two protein kinases, PKC and PKA. These protein kinases phosphorylate a stabilizing protein of the NAPI2A. It's called NAF1. So phosphorylation of NAF1 initiates degradation of NAPI2A, endocytosis of NAPI2A, and NAPI2A degraded in the lysosome. So PTH trying to reduce NAPI2A through the endocytosis. So signaling of the FGF23 is similar to the PTH, and FGF23 has a kidney-specific receptor as well, so which is complex, the crosso and the FGF type 1 receptor complex. So FGF type 1 receptor express ubiquitously, all tissue, all cells express the FGF receptor, but crosso is a tubular specific protein, and crosso determines the specificity of the FGF type 1 receptor towards the FGF23. So FGF23 activates the crosso FGF type 1 receptor complex and activate MAP kinase in this case, not the PKC and PK, and MAP kinase activate SGK1, and this is downstream kinase, but the same protein NAF1 is phosphorylated by SGK1 and induce the NAPI2A endocytotic protein degradations. The deletion of the crosso and the FGF23 pathway in the mice shows a huge phenotype. Like a crosso knockout and FGF23 knockout mice had a very, very short lifespan, about 60 days of life, compared with the 400 to 500 of the mice. So this shorter lifespan is largely due to dysregulation of the phosphate homeostasis. So FGF23 and crosso knockout mice develop hyperphosphatemia, increased serum phosphorus, and so they have an extremely higher level of phosphaturic hormone like FGF23 and PTH. So dysregulation of the FGF23 and crosso axis shows the total disruption, systemic disruption of the phosphate homeostasis. So due to the hyperphosphatemia, FGF23 and crosso knockout mice developed medial calcification, severe vascular calcification. So minerals like calcium depositing the aortic and the small arteries, the medial layer. That's why they have a shorter lifespan. In contrast to the knockout, the activation of the crosso FGF type 1 co-receptor extend the lifespan of the mice, So this is the two strain of the colossal transgenic lines. Both of the strain shows the extended lifespans. So they have a longer lifespan than the wild type mice in both males and females. So similar to the mouse, in this case, like FGF23 and the FGF23 and the colossal deficiency, the decline of the kidney function is associated with the dysfunction of the phosphate homeostasis. So reduction of the EGFR, decline of the kidney function, linearly increase serum phosphorus and FGF23 as well as the PTH. Whereas the colossal is linearly reduced by the decline of the kidney function, reduced EGFR levels in humans. So wrap up the background. So kidney is the central site in the phosphate homeostasis. So CKD and aging reduced the kidney functions, and which is reduced the colossal expression and result in an extreme increase in the FGF23 and PTH phosphaturic hormones, as well as serum phosphorus. So systemic dysregulation of the phosphorus induced vascular complications such as vascular calcification and cardiac hypertrophies. However, the player around the phosphate homeostasis are pretty low, about 7 to 8 players around. So we're thinking that more players should be involved, especially the protein selectively expressed in the kidney, because kidney is a central site in the phosphate homeostasis. So we are looking for the novel proteins that are regulating phosphate homeostasis, especially the protein selectively expressed in the kidney, and using RNA-seq and over 20 mouse and human tissue, we found that about 54 kidney specific genes, including 4 known factors involved in the phosphate, NAPI2A, NAPI2C, clotho, and the PTH type 1 receptors. But we don't know whether other 50 kidney-specific genes involved in the phosphate homeostasis or not. But the 50 genes are too much, so we cut down the list by analyzing RNA from the primary human proximal tubular cells and found that 18 proximal tubular specific genes. So we work on the 18 genes using the cherry-pick RNA library, and we successfully reduced all genes specifically expressed in the proximal tubular cell about 60 to 70%. So that assay is subjected to phosphate uptake experiment using the human proximal tubular cells. So FGF23 is an important endocrine hormone, downregulate phosphate uptake of proximal tubular cells in presence of the control RNA. But to make a long story short, we found that one kidney specific proximal tubular specific gene called TMEM174. Inhibition of these genes completely abolish the action of the FGF23, completely block the FGF23 mediated phosphate uptake of the proximal tubular cells. Similar to the phosphate uptake, TMEM174 SI treatment completely block the reduction of the NAPI2A by the FGF23. So FGF23 reduced the NAPI2A protein proximal tubular cell, but which is totally blocked by inhibition of the TMEM174. This is a confirmation whether TMEM174 involved in the FGF23 and the PTH mediated NAPI2A endocytosis using the human cells and opossum proximal tubular cells. Both FGF23 and PTH reduced the NAPI2 protein in time-dependent manner through the endocytosis. But in presence of the TMEM174SI, FGF23 and PTH no longer reduce NAPI2A proteins. So TMEM 174 somehow involved in FGF23 PTH mediated phosphate homeostasis through the NaPi2a regulations. So TMEM174 is unfunctional proteins, but as I said, specifically expressed in the kidney and are selectively expressed in the proximal tubule of the kidneys, and especially the expressed in the apical membrane of the proximal tubular cells, because the TMEM174 co-localized with the apical membrane marker LTL. So all the green, red is co-localized green, it makes yellow. And TMEM174 is a type 3 transmembrane proteins, very small protein around, they consist of around 240 amino acids, about 25 kilodaltons. So TMEM174 is located in the plasma membrane and co-localized with NaPi2a, and more importantly, TMEM174 directly bind to NaPi2a protein using the IP analysis. This is an overexpressing experiment. So overexpression of the flag NaPi2a pull down the TMEM174, but not stabilizing protein NaPi2a NAF1. So we next generate the TMEM174 knockout mice. This knockout mouse grew normal, and they had a normal kidney function level of serum creatinine that's similar, and also they have a histologically normal kidney. And electrolytes like calcium, magnesium, sodium, potassium, chloride are all similar between wild type and TMEM174 knockout mice. But the factors involved in the phosphate homeostasis shows huge differences. TMEM174 knockout mice developed hyperphosphatemia. Also, they had an extremely higher level of FGF23 and PTH, like a patient with CKD. And whereas active vitamin D levels are pretty similar between the two groups. So hyperphosphatemia by TMEM174 deficiency is due to increased retention of the NaPi2a, whereas the interestingly NaPi2c, another proximal tubule specific phosphate transporter, really reduced. We're thinking this reduction is a compensation of the increased extreme induction of the NaPi2a. But NaPi2a is a major one to reabsorb the phosphate in the proximal tubular cells. So increased NaPi2a result in increased phosphate uptake of the brush border membrane of the kidney in sodium dependent manner, but independent sodium uptake no difference between the two groups. So NaPi2a is a reabsorption phosphate transporter. Therefore, the TMEM174 knockout mouse had a significantly higher urinary excretion of the phosphate and result in hyperphosphatemias. So no calcium urinary excretion or no difference between the two groups. So since they developed a severe hyperphosphatemia, TMEM174 developed a severe medial calcification as well. So a lot of calcium accumulated in the otic media. And we're also analyzing the flexibility of the mouse aorta using the new system that we are purchased, flow velocity system. This is really good system to analyzing small rodents of the flexibility of the blood vessels. So two probe can detect the either coronary artery signaling or the abdominal signaling. Using that system, basically TMEM174 deficiency increases vascular stiffness. They're losing flexibility of the blood vessels. So I want to wrap up my talk with this slide as a conclusion. So PTH and FGF23 bind to the receptor in the proximal tubular cells. and activates their receptors, and somehow TME174 are activated through the unknown mechanism, but this is a NaPi2A binding proteins and initiates destabilization of NaPi2A NAF1 complex and induced endocytosis to reduce NaPi2A protein to block the phosphate reabsorptions. Thank you.
Okay, thanks very much. Interesting presentation, Makoto. Do you have any information about any of the signaling pathways affecting NaPi2B with respect to regulating phosphate absorption from the GI tract as opposed to just being a function of the intake?
NaPi2B is not affected by TME170 because my protein is just only specific kidney, but I think that most of the signaling through the intestinal NaPi2B is vitamin D.
Is by what?
Vitamin D.
Okay. And what about treatments that the nephrologists use to treat hyperparathyroidism and reduce levels of stimulation? Does that have the appropriate effect?
PTH has a kind of so much effect, so might be more better to target the FGF23, which is more specific to the phosphate. So yeah.
Okay, interesting. All right, let's see if I don't have any other questions here. So good. Thank you both very much for your presentations. I appreciate the work that went into you getting ready for it. So thank you very much.
Thank you.
Bye-bye.
Thank you. Bye.